Hello, everyone. This is Mr. Friedhoff. I'm going to be going over the 12th chapter with you. This is going to be a continuation of what we learned in the last chapter when we talked about DNA. And we're going to talk about in this chapter why DNA is important and what it actually does. We know that DNA codes for our physical characteristics, but in this chapter, we're going to explore how that actually happens at a deeper level. So, with that being said, our first section is going to be just about the central dogma. This is that flow of genetic information going from DNA to RNA to a protein. These proteins are going to be a part of our phenotype or our physical appearance. So again, we're just looking at how DNA is going to code for our physical appearance. There are three main parts of the central dogma. The first part is the replication, which we already went over. That's DNA copying itself. Then we're going to talk about transcription, and lastly, we're going to talk about translation. These processes are all in the central dogma of molecular biology, or just biology in general. This dogma means just a kind of like a set of rules or guidelines that life follows. All life on Earth follows this dogma or this genetic information going from DNA to RNA to a protein. So it's really important that we understand this one slide. This one slide really encapsulates basically all of biology. All right, so we have a different type of molecule that we're going to learn about in this chapter. Specifically, it's called RNA. This is called ribonucleic acid. There are several different functions. You can see there's like mRNA, tRNA, and rRNA. Basically, what we're going to be looking at in this chapter is how these RNA structures are going to help us facilitate something called transcription and translation. But what I want you to know about RNA is it's very close to DNA, but there's a couple small differences. First, it's single-stranded, so it means it's only one strand of nucleotide. Second, we have a different type of sugar molecule. In DNA, we had deoxyribose. In RNA, we have just regular ribose. Lastly, RNA contains a base called uracil instead of thymine. So you can see here this thymine is going to be replaced with uracil. There are several differences between DNA and RNA. They're going to enable RNA to do different things. So the reason why we have RNA is to kind of facilitate different reactions, and this RNA or the differences within RNA allow that molecule to do different things than DNA does. Here's a different look at this RNA versus DNA. You can again see this double-stranded DNA here, the single-stranded here. You can see RNA has uracil while DNA has thymine. All right, hello everyone. This is going to be the second section of chapter 12, and this is where we're going to talk about transcription. Now, transcription occurs in the nucleus, and it's going to be where a molecule called RNA polymerase is going to unzip and copy the DNA. Now, RNA polymerase is an enzyme. You can see it ends in this ACE, but RNA polymerase is just going to unzip the DNA, and it's going to make a copy of that DNA. And that copy is going to be called mRNA. This is that process. You can see the mRNA being produced here. It's in red. This is green feature here. That green structure is the RNA polymerase. And you can see it's matching up base pairs just like we saw in DNA replication. However, in this case, we're using RNA, so we don't use thymine, we use uracil. It still has the same bonding base pairing, C's with G's, A's and U's bond together. But it's just a little bit different. This is called transcription. So again, you can see what this looks like. You can see RNA polymerase bonding to a region of DNA. It's going to transcribe that DNA. And basically what we're doing is we're making a copy of that DNA. And that DNA is the genetic code of life. So again, we're just making a copy, but we're using RNA. You can see that RNA polymerase, it binds to a location called the promoter, and it's going to transcribe a gene until it reaches the terminator site. That terminator site is going to be a location where the RNA polymerase unbinds. And then this RNA here, this mRNA specifically, is going to travel outside the nucleus and do other things. Whenever we transcribe DNA, it's important to remember that DNA has thymine, however, RNA has uracil. So A bonds with U, T bonds with A, G bonds with C, and C bonds with G. So if we look at this line here of DNA, it's going to be C, G, U, A, U, C, C. You can see everything binds pretty much the same as replication. However, remember in RNA, T is replaced with U. If I had a choice between DNA and RNA, I would choose RNA because it has a U in it. All right, that's the end of 12.2. Hello, everyone. This is going to be the third section in unit or chapter 12. This is going to be about translation. So in the last section, we talked about how DNA is transcribed into RNA. Now we're going to learn that translation is a process where we take that code in mRNA and we make a protein. So we're basically translating this code of RNA molecules into a protein. Remember, we're talking about the central dogma of molecular biology where we go from DNA to RNA to a protein. And this genetic code in our DNA is actually going to code for the protein. However, we're not going to use that DNA code directly. We have to make a copy of it using RNA. And that specifically is going to be then translated into a protein. And then these proteins are going to code and give us our physical features. So in this section, we're going to learn what that actually or how that actually is accomplished. Remember that proteins are made of amino acids, and each protein has a bunch of amino acids. Now, there are 20 different kinds of amino acids, and you can see this in our chart here. Each amino acid has a 1- and 3-letter abbreviation. If you look, you can see here, here's our amino acid list, here's their 3-letter abbreviation, and here's their 1-letter abbreviation. So again, our proteins are made of amino acids. There are 20 different types of amino acids, and they have a name, a 3-letter abbreviation, and a 1-letter abbreviation. Now, when we look at this codon, or we look at this mRNA strand, it's going to be made of codons, and codons are 3 nucleotides in a sequence. You can see again, here's a codon, here's a codon, here's a codon, here's a codon. Each of these codons is made of 3 nucleotides, and specifically, they are RNA nucleotides. So each codon is then going to code for 1 amino acid. You can see each mRNA strand has many of these codons. So each codon is made of 3 nucleotides, and each codon is going to code for 1 amino acid. Here is a codon chart. This codon chart is going to allow us to figure out what amino acid each codon makes. So you can see here, we're going to start in the middle where we have our first letter. Our second layer here is the second letter. And the third layer here is the third letter. So this is, again, a codon chart that we're going to go through, and we're going to figure out what the amino acid is that codes for each codon. So let's do an example here. So again, we're going to start here in the middle, and we're going to work our way out. For the first one, GCA. If you look here, we're going to start with G. We're going to then go to C. Then we're going to go to A. You can see that's going to code for the amino acid ALA, or A. This is alanine. Next one, CAU. So you can see CAU codes for histine, HIS or H. UAG, you can see UAG. Oh, there's a little square here. That's going to be one of our stop codons. You can see UAG makes a stop codon. AUG, this is our start codon. We're going to learn about those next. Every protein is going to start with a start codon, and it's going to end with one of these stop codons. Here's another way of looking at it. You can see this is another codon chart. It's a little bit different. Let's do this again. GCU. So we're going to start here. GCU. That makes alanine. So you can see I went one. Here's our first base. Here's our second base. And then our third base. So again, GCU makes alanine. Then like UUU. So you can see UUU. Phenylalanine. CGA. C on this side. We find G and A. Arginine. Last one, AGG. So we're going to start AGG. That's another arginine. Good. So again, we're going to transcribe our message. You can see our first one here. So again, our RNA has the U's. This is the transcription. Then we're going to translate. So you can see CGU makes start. AAU makes ASN. CCG makes PRO. UAC makes TYR. And UGA is our stop codon. So each one of these 3-letter abbreviations stands for some type of amino acid. We can see we start out with the start codon and we end with a stop codon. So just like I said, every protein is going to begin with a start codon, and every protein is then going to end with a stop codon. There are 3 stop codons, UAA, UAG, and UGA, while there's only 1 start codon. It's very cold outside today. It has been really cold. Make sure you have your coat on. Get it like coat on, like a coat. Okay. This is going to be end of 12.3. Here's my analogy for protein synthesis. Say we have this cookbook in our library, and this cookbook has instructions to make a cake. We don't want to take this book out of the library because it's very special. It's a very special book. We don't want to get damaged. So what we do is we make a photocopy of this. Now, this photocopy is going to allow us to read the instructions at home. However, again, the analogy here is that the original book is like DNA, and our photocopy or the method of copying it is like transcription. The photocopy itself is mRNA. So we're making a copy of the DNA, or we're making a copy of the original instructions, and it helps protect the original instructions so we can use them again. Then we go home and we read those instructions and we make our cake. This is translation. This is going from this copy of the genetic code to the actual protein that we want to make. So this is an analogy between DNA, RNA, and protein, and making a cake using a set of instructions from a cookbook. All right, 12.4 is going to look at a little bit more of what happens in protein synthesis. This is where, again, translation occurs. So specifically, we're looking at translation. What happens is mRNA is going to leave the nucleus and travel to the ribosome. So you can see here's our ribosome, and what happens is the ribosome is going to read this genetic code. And how it does this is it just runs along the mRNA strand. Now, what this ribosome will do, as you can see, is this ribosome will start adding on these tRNA molecules, and these tRNA molecules will bring in these amino acids. These are going to correlate to the amino acids that we just learned about. So what happens here is you can see this ribosome is going to make our protein chain. Now, this protein is made first of something called a polypeptide. This is just a long chain of amino acids. But basically what we're just showing you is a more complicated version of what protein synthesis looks like. So you can see our ribosome attaching to the mRNA. These tRNA molecules are going to bring in the amino acids, and those amino acids are going to start bonding together, and they're going to start forming things called peptide bonds. So between each one of these amino acids is a peptide bond that's going to make the longer protein just in general. Now, this translation reads, like I said, translation is going to read the mRNA strand. The tRNA molecules bring in the amino acids, like I mentioned, and these are what is going to actually facilitate the process of translation. These amino acids are going to also bond together to form a protein. Now, just a little bit of just kind of more information. There is RNA before and after the start and stop codons. I don't show you that in our worksheet that I go over because I just don't want you to kind of get confused. But just understand there's more RNA before and after that helps kind of protect the inner parts of this DNA. It helps it from beginning, it doesn't get damaged. All right, next we're going to talk about 12.5, which we're going to talk about why genes produce proteins and what the importance is. This one slide is probably one of the most important slides of the whole year, where we're going to look at how genes are going to code for proteins that make us who we are. So again, proteins are what determines our physical characteristics. And our proteins, again, make us who we are. Now, these proteins are from our genes, and our genes are from our chromosomes. And these chromosomes are from our parents. So this is why you look the way that you do, is you inherited chromosomes with DNA that make proteins and make you who you are. So again, going from a gene to a protein, genes are found on chromosomes. These genes make proteins, and these proteins make us who we are. Now, the amount and order of these amino acids are going to determine the shape of the protein. The order of the amino acids is like the order of letters in a sentence. How we arrange the letters in a sentence makes that sentence what it is. So those letters are going to be arranged in a sentence to make the sentence mean something. Same thing with amino acids. These amino acids are going to be arranged in the protein to make it actually do something. Now, what will happen is these amino acids will start bonding together and start folding together to make some shape. That protein's shape is going to determine its function. You can see here, these are just different amino acids, and how they're going to interact with each other is going to determine the structure of that protein. So the order of which of these amino acids is going to make the structure of the protein. The shape or structure is going to determine that protein's function, thus making us who we are. Here's a video of what that kind of looks like. So you can see here, these are just the different amino acids. They're going to start kind of interacting with each other, and they're going to start forming some shape. And that shape is going to determine that protein's structure. That protein structure is going to have some phenotypical characteristic that makes us who we are. So you can see these amino acids are going to make this shape, which is a certain type of protein. All right, this is going to be our last section in chapter 12. This is going to be on cell differentiation. This is the process by which cells are going to become more specialized. So think of cells as when they begin, they're actually going to be known as stem cells. They're kind of undifferentiated. They don't have a specific job. What will actually happen is certain genes will be activated, and those genes will determine what type of cell it will become. So you can see here a single cell can either become like a neuron, an epithelial cell that's like a skin cell. It can become a host of different cells. But each cell has a whole genome inside it, the whole set of instructions to make whatever cell that it needs to. What will happen is certain proteins will be activated, or certain genes to make certain proteins will become activated to make it a certain cell. So sometimes we want cells to be neurons, sometimes we want them to be epithelial cells, sometimes we want them to be stomach or eye cells. It depends on the organ in which they are found. When those cells are undifferentiated, we call those stem cells. And these stem cells will become some type of cell. They can become like a muscle cell, a neuron cell, an immune cell, but it will be based on what genes are going to be active or inactive in that cell. This occurs during your development. Cells will differentiate and become specialized to perform different functions and tasks. But again, that cell's shape is going to be determined by all the proteins that are active in it.